Hello, I'm very pleased to give you the update for the MSA Collaborative Cause Program 2022. My name's Vic Khurana. I'm a movement disorders neurologist and a physician scientist at Brigham and Women's Hospital and Harvard Medical School in Boston. I'm also the scientific liaison on the board of directors of the MSA Coalition. I do not have any relevant disclosures for this talk. Some of you will remember from prior years that we've described the Collaborative Core program before. It's really one of the flagship programs for the MSA Coalition's Research Committee. The inception of this program really dates back to key questions that were raised at a meeting of various stakeholders, including clinicians and scientists, advocacy groups, and so forth, at the Global MSA Research Roadmap meeting in Las Vegas in 2014, 8 years ago now. And at that meeting, the field was asking what key questions would need answering in the near future to really shift the needle on this disease for our patients. There are many mysteries about MSA, but one of them that comes up all the time is what causes this disease. It's incredibly frustrating. Any patient who comes to see me with this disease has this question, why me? How did I get this? What causes this disease? And at this time, I cannot give them a clear answer. We think that like many complex diseases, Alzheimer's disease, Parkinson's disease, diabetes, there's a combination of genetic and environmental factors that causes MSA. And so that's about the best we can do right now without any of them listed. So that was defined as one fundamental question. Another question, in the brain, we understand what the pathology looks like at the end stage once a patient has passed away. We've examined the brain after death, a post-mortem examination. We have become quite good at identifying the hallmark pathologies of this disease and diagnosing this disease from the post-mortem tissue. But that doesn't really tell us how the disease started. That's an end-stage brain. What happens at the beginning of this disease? Which cell in the nervous system is the culprit? And how can we model this disease in the lab? How can we model it in a productive way such that insights in the lab actually answer questions for patients? And how can we model it so that if we develop a treatment or a cure or a way to slow this disease down in the lab, we have a pretty good shot at that actually working in real patients. Thirdly, what drives the progression of this disease and how do we measure it? You've probably heard the term biomarkers. Biomarkers are ways in which we can tell that the disease has started and a way in which we can tell how quickly the disease is progressing. Biomarkers include factors that we can recover from blood tests or from spinal fluid or from imaging the brain, to name a few examples. And often we have to create biospecimen repositories in order to establish these biomarkers. And so this third question that was raised in this meeting was related to how we would in practice achieve that understanding. And finally, of course, how do we treat this disease? In some ways, that relates to the preceding three questions. And there are two types of treatments that were thought about. Symptomatic treatments to make patients feel better, and also disease-modifying therapies. Treatments that could either prevent, reverse, slow down this disease. So these were the questions that were posed, and the Collaborative Cause program was really a response to these questions. And so in response to the genetic and environmental question, we have developed ideas for Core G and Core E. G for genetics, E for environment. In order to think about where this disease starts and how to model it in the lab, Core M, M for models. Biomarkers and biospecimen repositories, Core B, and the SEED program of the MSA Coalition that many of you are familiar with has continued to thrive and support new ideas related to therapeutics of MSA. So in a little bit more detail, let's run through some of these cores. So there are two cores that have actually been funded. Excitingly, in the last couple of years, this has happened, and we're thrilled about this and extremely grateful to our supporters who've made this happen. Core G for genetics. So in Core G, we're collecting genetic information from a diverse group of MSA patients to better understand the genetic causes of MSA and uncover new therapeutic targets. In Core E over here, we're developing an infrastructure to register environmental exposures in MSA patients to better understand environmental triggers for the disease. There are two cores yet to be funded. These are Core B for biomarkers. In this core, we hope to assemble biomarkers from patients to aid in the early and specific diagnosis and to recruit and track outcomes in clinical trials. In Core M, down at the bottom here on the bottom right, we are building next-generation models of MSA for drug discovery using cutting-edge human stem cell technologies. So let's talk a little bit about the funded cores. Core E is really supporting the pioneering work of Beata Ritz at UCLA. Now, Beata has spoken at a conference previously, and you'll be able to get a copy of that recording. That work continues. She's been a pioneer in understanding the environmental causes for Parkinson's disease, and we hope that the exciting program that she's developed for multiple system atrophy will do the same for MSA. The challenges, of course, whether it's Core E for environment or Core G for genetics, is that MSA is a very rare disease, and it will be very challenging to establish the numbers of samples we need to get definitive answers. But we need to start somewhere. We need to bring this information together from across the world, from different centers, and arrange that information properly. And for environmental exposures, Dr. Ritz is really leading the charge. And we think that this is a very important endeavor for our patients. Many of our patients ask us whether certain things they may have been exposed to in the environment may be causing this disease. We'd like to be able to give them definitive answers. Now, Core G is an exciting international core, and it comprises a number of different scientists. It is, like all of the cores, designed to complement what's already going on in the MSA community and to bring together information from across the world. So in this case, let's move from left to right here. Dr. Farah at the University of Florida is leading the genome sequencing efforts in Core G and identified for the coalition hundreds of DNA samples that were actually in Korea that had no other mechanism of being sequenced. It's incredibly important that we create diverse specimens. And so to complement ongoing MSA sequencing efforts at the NIH, we are leading the charge on sequencing genomes that would otherwise not be sequenced, including these precious genomes from a Korean cohort of MSA patients. Sequencing with Dr. Farah's group and Dr. Huntelman's group also include European samples from the United States and Canada. We are collecting genetic information that's already been assembled at the University College London by Dr. Henry Holden, and we're bringing that together with these other genomes. Now, we need to bring this all together in a useful format for the community, and Dr. Dana Vuzman at Harvard Medical School is leading this effort. Dana leads the clinical genome analysis platform at Harvard Medical School, and she's closely advised in that endeavor by a very senior geneticist, Dr. Shamil Sanayev, who's taken a strong interest in MSA as well. And a postdoctoral fellow, Dr. Samaya Nazeen, is helping this effort by developing algorithms for the analysis of these genomes. So what CGAP, the Clinical Genome Analysis Platform at Harvard, is planning to do is amalgamate these different genomes and genomic information from these various groups and assemble them in a coherent way that is searchable and made public to the community, researchers and patients alike. And this is a very large effort, and I would encourage you to listen to Dr. Vuzman's talk on Core G this year at the conference. She'll be giving a lot more detail about the exciting progress that she's made. Last year, I spoke about the scientific basis of Core M. It's centered on a technology known as iPS cells, induced pluripotent stem cells. For a complex disease like MSA, it makes sense to create not just mouse models, but human models of the disease. So far, any treatment that has worked in a mouse has not worked in clinical trials of human beings with MSA. Now, there's many reasons for that, including our deficiencies at recognizing the right types of patients and running the right types of clinical trials. Other cores, like Core B, would be instrumental in solving that problem. But we don't have great models of this disease in the lab, and so it makes sense to try and push the boundaries there. Fortunately, there is this technology that allows us to take a patient with the disease, take a skin biopsy, grow skin cells from that biopsy, turn them back in time to these induced pluripotent stem cells that are embryonic stem cell-like cells from which we can generate any cell in the body, including nervous system cells, neurons, glial cells, which are critically important to the neurodegenerative process that occurs in MSA. So we can effectively make a patient-specific model in the dish in the lab. And we can even incorporate these cells now into mice so that human cells are growing within mice, and we can study human biology in the living animal. This is really a remarkable development, and we hope to be able to exploit this in Core M. Now, there's been some exciting progress in. Now, last year, you may recall if you attended the talks, that I did talk about establishing an iPSC and brain bank for Core M. And this work actually started some years ago in 2014 with a coalition grant to my group. And I'm thrilled that in 2022 we can announce some real progress here. So the aim was to build a small number of iPSC lines from well-characterized patients with MSA matched to post-mortem brain. Why did we want to do that? Well, if we're going to make these exciting new models, we need to make sure they're relevant. These very generous patients who donated their brains after death have enabled us to generate these matched iPS lines to brain so that when we do make these models, we will be able to ensure that they faithfully recapitulate what's happening in the patient brain of the exact same donor who gave us those cells. So, as I mentioned, there's been progress in 2022. Now we will release to the MSA research community, thanks to multiple stakeholders, including our colleagues, patients, and their caregivers, iPSC lines matched to post-mortem brain. In a forthcoming publication, we'll describe 38 fibroblast skin cell, or iPSC lines, from patients, 22 of whom had MSA, and others were so-called controls, either with related diseases or who were healthy controls. And 15 of those MSA lines will be matched to post-mortem brain. So this is, we hope, a great resource that will be released to the community. This effort has been led by Alain Ndayisaba at Brigham and Women's Hospital, and he is closely supported by senior mentors Gregor Venning and Nadia Stefanova at the University of Innsbruck. This work would not have been possible without the seed grant from the coalition, and like all good seed grants, it blossomed into a project that was worthy of funding by multiple others, including, over time, the Fox Foundation, the Aligning Science Across Parkinson's Initiative, Brigham and Women's Hospital, the New York Stem Cell Foundation, and the NIH. So in the last few minutes, I'd like to explain how Core-M can crosstalk with the other cores. So I'd like to tell you about a patient S, who was one of my patients, and she comes from a remarkable family. She had 6 siblings alive at the time when we saw her, and 2 parents who were nearly 100 years old. Remarkable to have that many people in one family to analyze the genomes. And so we decided to deposit her DNA into Core G, the genetics core. And Matt Huntleman in Core G studied her in the context of a study for families just like this with MSA. And this is a picture of this woman's pedigree. This is her over here. These are her siblings, and these are her 2 parents. Her parents by now have both deceased, and the patient herself is deceased. This was some years ago when this was started. And what we discovered was that she had several interesting changes in her genome, and one of those genes had previously been identified in our lab as involved in the toxicity of the protein that really is central to MSA and Parkinson's, called alpha-synuclein. So this was a really interesting convergence of data, genetic data with cellular data from our lab. And now through Core G, this genome of this patient and her family is being intensely studied and analyzed on the CGAP platform by Dr. Vuzman and her colleagues, allowing us far more detailed genetic analysis. And what we'd like to ask is if some of the interesting genetic changes in this woman are not just unique to this family, but are also present in other patients with MSA. And this allows us to ask a very specific question, and Core G is really facilitating that. We are now, in the next year and a half, going to be able to ask that question in genetic information from the University College London, from the Korean genomes, from our collaborators in Japan, and also AMP MSA, led by Sonia Schultz. And if we can corroborate this, we can return back to Core-M, where human stem cell models that have already been made from this patient can be thoroughly analyzed, and not just in a generic way, but in a very specific way. We can ask very clearly if the gene that we have found is really important in triggering disease-like changes in the stem cells that we make from this patient. So this is one way where Core G and Core-M can communicate. Now there's other crosstalk that's going to be possible. When Beata Ritz, for example, identifies toxicants in the field that may be related to MSA, human stem cell models that we make in Core-M will be able to address critical questions. And there's a precedent already established by Parkinson's disease research. This can give us a roadmap. For example, Beata, along with Kimberly Paul, a professor also at UCLA, have performed quantitative assays of environmental toxicants and related them to Parkinson's disease risk. They've assembled a group of toxicants that they think are potentially involved in Parkinson's. And they've teamed up with collaborators, including Lee Rubin, Rich Krolewski at Harvard University, Edinson Lukumi at Brigham and Women's Hospital, to test this out in brain cells, in this case dopamine neurons that are important for Parkinson's disease, in the laboratory setting. So now a screen in humans can be brought to a screen in the lab in brain cells made from patients. And this has used some very innovative technologies for generating pure populations of these dopamine neurons. The findings from this study have been really informative so far, and in particular have highlighted combinations of toxicants that are particularly potent in triggering dopamine neuron death. This will lead to studies identifying how these toxicants are toxic, but also, we hope, to public health changes that will prevent these toxicants from being used in the future and hence decrease the risk of terrible diseases like Parkinson's and MSA. So I hope I've given you a flavor for how some of these exciting cores like core M and core B can be added to the existing cores, core E and G. We hope that these, with your support, are funded in the near future. We certainly can't do this alone. We'll need a team effort. We'll need to partner with you, and thank you again for supporting our efforts and for your attention. This story will be continued. Thank you again.